Today we'll be talking about cloning human organs. Before we begin, there is some concepts that we'll need to understand. The first thing that you need to understand is how the organs are being cloned. This is done through a process known as somatic cell nuclear transfer. This involves removing the nucleus from a healthy egg cell. Scientists then transplant the nucleus from a donor's somatic cell into the egg cell, which is then artificially activated to make an embryo. The next concept that you'll need to know is the types of cloning. The first type of cloning is called therapeutic cloning. This involves cloning which produces embryonic stem cells with the same DNA as the donor cell. The next type of cloning is called reproductive cloning. This is implanting the embryo into a surrogate mother to produce the cloned organism. The last thing you'll need to know is some definitions of words that will be used throughout this video. The first word is enucleated eggs. These are eggs with the nuclei removed. The next one is embryonic stem cells. These are a stem cell that is pluripotent, meaning it's capable of taking the form of multiple cell types. Blastocyst. This is the stage when the embryo contains around 150 cells after fertilization. The next word is xenotransplantation. This is the process of transporting animal organs into humans. The last one is differentiation. This is the process of embryonic stem cells maturing into body cells. Now we shall show you a video re-explaining these concepts.
In 1996, the first clone of an adult mammal, a female sheep named Dolly, was born. To create Dolly, scientists used somatic cell nuclear transfer, or SCNT, a revolutionizing technique that now is a cornerstone of stem cell research. In SCNT, scientists begin by removing the nucleus from a somatic or body cell of a donor mammal. They then collect an egg cell from a donor of the same species. The egg cell nucleus is removed and replaced with the nucleus of the somatic cell. Once inside the egg, the somatic nucleus is reprogrammed by cytoplasmic factors in the egg, returning it to an undifferentiated embryonic stage. The egg may then be implanted into the uterus of a surrogate mother to produce a cloned animal through a process known as reproductive cloning. Alternatively, it may remain in the laboratory to be used in studies of therapeutic cloning. In therapeutic cloning, the fertilized fused egg cell is allowed to develop to the blastocyst stage. From the blastocyst's inner cell mass, scientists produce a culture of embryonic stem cells, ESCs. Through the process of differentiation, ESCs develop into any one of the many different types of mature body cells that make up an organism.
Now we shall move on to the article. The article we'll be going over today is Could We Clone Human Organs to be Used in a Transplant? Published in 2008 from How Stuff Works. The article addresses 5 key topics, and the first of these topics explain how human cloning works, which is the previously aforementioned somatic cell nuclear transfer. The next topic that the article addresses is the current status of human cloning during 2008. In the article, it mentions how research into human therapeutic cloning has come mostly to a halt in the USA. This is due to 2 major factors, one which is the bioethical issues of human cloning. The other major reason is due to the lack of available human egg cells that are available for research. This is because laws and ethical regulations from the National Academy of Sciences and the International Society for Stem Cell Research prohibit monetary compensation for females who donate their eggs for embryonic stem cell research. Coupled with the low rates of success for embryonic stem cell research, an abundance of eggs is required to make progress for this new technology. That is why Ian Wilmot, the person who cloned Dolly the sheep, has suggested that they could use animal egg cells as a substitute for human egg cells. The next topic that the article addresses is the previous advancements from therapeutic cloning in animal studies. In March 2008, researchers removed skin cells from mice with Parkinson's disease, which is a brain disorder, to test a way to use stem cells as an effective treatment. The scientists then inserted the DNA from the skin cells into enucleated eggs and created cloned mice embryo. After extracting stem cells from the cloned embryos, the researcher developed autologous dopamine neurons from them, which are the nerve cells affected by Parkinson's disease. After implanting the new neurons into the mice, the test animals exhibited signs of recovery, showing the potential of how therapeutic cloning could be a treatment in the future. The next topic that the article addresses are the alternative options instead of therapeutic and reproductive cloning. These alternatives are xenotransplantation. which involves transplanting animal organs into humans. The other alternative is to get stem cells from adults and to regulate the 4 genes that influences the cell differentiation. Then the scientists reprogram the cell to become pluripotent.
Which demographics will be affected by the discovery? Here I'll be discussing the individuals, groups, ages, and genders that will be affected by the discovery of human organ cloning. Firstly, let's note that the main demographic being affected would be those who are in need of an organ transplant, maybe due to a disease or simple organ failure. According to the graph being displayed, there are approximately 97,200 people on the waiting list for a kidney transplant. These statistics display that the organ demand is high, whereas the number of donors of the organs are low. Individuals who have diseases which causes organ failure will be positively affected by the discovery of human organ cloning. Some of these diseases being diabetes, obesity, renal disease, and many others. The aforementioned diseases are commonly seen worldwide. The most common organ that fails would be the kidney. The effect of this discovery to the individuals and groups that have these diseases would be the mortality rate of said diseases lowering due to the supply of organs. Gender. For the sake of simplicity, I'll be referring to the 2 sexes assigned at birth, male and female. Men are more susceptible to organ failure compared to women. I'll be using kidney failure as an example because it's the most common type of organ failure. Men have a higher risk of kidney failure due to their difference in hormone levels compared to women. High testosterone levels in men may cause a loss in kidney function. Men's kidneys may not be protected by estrogen, which is high in women until menopause. This discovery will benefit both males and females. However, the benefit to males is slightly higher due to males having a higher chance of organ failure. This graph displays the number of organ transplants that have occurred in America, sorted by age groups. The statistics display that the elderly have the most amount of organ transplants compared to the other age groups. This is due to a multitude of reasons, though the main contributor would be the weakening of immune systems of the elderly. An example of a disease that affects the elderly, which in turn requires them to get an organ transplant, would be renal end-stage disease. According to an article written by Johns Hopkins Medicine, those aged 65 and older make up over 1/2 of the people with end-stage renal disease in the United States. End-stage renal disease is a scientific term for kidney failure. It is due to a gradual loss of kidney function due to aging, a condition, or an issue with your body. The discovery of cloning organs will greatly affect the elderly. As we can see from the statistics displayed in the article, only 10% of patients between the age of 65 and 74 are on the waiting list. This is because organ transplants are prioritized for those who are younger and guaranteed to live a longer life. Due to the short supply of kidneys and donors, the elderly are left to wait until they have an available donor. Now I'll be discussing the major issues raised from the topic, specifically legal, economic, health, environmental, social, and ethical. Firstly, legal issues. Australia used to have a ban on therapeutic cloning research up until 2006, where the ban was lifted on the condition that the research would receive no funding from the government. The article where the information came from does not elaborate on why therapeutic cloning was banned, but it is assumed to be because of ethical reasons. As for economic issues, there are none that are major and worth mentioning. Health. There are no major issues raised from the topic, rather the opposite. There will be major benefits if organ cloning is perfected. It will save many lives. Although at the current stage of therapeutic cloning, there are some health issues, some of which are there is no guarantee that the replaced organ will be functional. This could be due to many factors. One would be the process of cloning the organ was unsuccessful, making the organ defective. Mutations occurring when cloning stem cells are possible. These mutations have been observed to behave like cancer cells. Cells could be contaminated with viruses. However, once these issues are resolved, there will be no health risk at all. Environmental. There are no environmental issues raised from organ cloning. Social. As for social issues, we would have to educate the public on how the cloning process works and whether or not it is ethical, and we also have to justify why we are performing unethical experiments on organ cloning. And finally, the last one, ethical. By following ethics, researching cloning is wrong for many reasons. The first being, when you clone, you're practically creating a living cell, then destroying them for research. Ethics can argue that this is abuse and mistreatment for a living being. There is also the question of, at what stage of its development should an embryo be regarded as and treated as a person? And the last question would be, should embryos come to be viewed as a commodity and not as an embryo that could develop into a person? These are all ethically correct and justified, but can be solved by convincing the public that their unethical practices will benefit humanity.
Questions about cloning. 1. Once cloning technology is perfected, will you be able to create a custom human by combining cloned body parts and organs? 2. Once cloning technology is perfected, what rights will human clones have, and how will society react to these cloned humans? 3. Once cloning technology is perfected, will it be possible to revive back people who are deceased?